All glories to God. Welcome back to our Kalanchiamedai channel, easy to understand biological terms. This video we will see the gluconeogenesis. As biologists, we are well known that each and every metabolic activity in our cell needs a glucose as a fuel. But the supply of glucose is not regular because between the meals or during the fasting, the glucose supply is interrupted. During that time, our cell undergo the process of gluconeogenesis. The gluconeogenesis means the conversion of glucose molecules from non-carbohydrate precursors, for example, pyruvate, alanine, or some of the amino acids. This gluconeogenesis process, the definition of gluconeogenesis process is the synthesis of new sugar. Yes, the synthesis of glucose from non-carbohydrate precursors. How our cells are synthesized? The glucose from non-carbohydrate precursor, let me see that process. In glycolysis, through the glycolysis only our cells are getting ATP, that's called the energy from glucose. But in gluconeogenesis, our cell release the energy from non-carbohydrate precursor. This gluconeogenesis is a step. It is a reversal steps of glycolysis. The 7 steps are same in glycolysis and also gluconeogenesis, but in reverse manner. The 3 steps are non-reversible. Our cell, especially in human, the gluconeogenesis process takes place in liver, not in muscle and some other cells, because the muscles or the brain cells lack certain enzyme, which mainly occur for the gluconeogenesis process, so that our liver convert the glucose or obtain glucose from the non-carbohydrate precursors. The 3 steps of glycolysis is bypassed in gluconeogenesis. Let me see what are the steps are bypassed in gluconeogenesis. The first step is the conversion of pyruvate to phosphoenol pyruvate. It is the first bypass reaction. During this gluconeogenesis, the both cell organelles are involved. We know the glycolysis only takes place in the cytoplasm, but the gluconeogenesis process occurs in both mitochondria and also cytoplasm, because it needs a mitochondrial enzyme. That is the first enzyme which is involved in the gluconeogenesis. That enzyme is called pyruvate carboxylase. This pyruvate carboxylase enzyme only present in the mitochondria. So the pyruvate molecule which are converted from the lactate are directly in the mitochondria. That directly transferred to the mitochondria. If the lactate is a precursor for gluconeogenesis, it converts pyruvate and is sent to the mitochondria. Pyruvate is directly present in the mitochondria that are involved in gluconeogenesis. The first enzyme is pyruvate carboxylase. This pyruvate carboxylase is a mitochondrial enzyme. So, from this pyruvate carboxylase enzyme, this enzyme first converts a pyruvate into oxaloacetate. For the functioning this enzyme, the coenzyme biotin is involved. This coenzyme biotin involved, that coenzyme biotin convert the pyruvate into oxaloacetic acid and also transfer one phosphoryl group. It is phosphorylated. Then the pyruvate molecule becomes a oxaloacetate with the help of the bicarbonate ion. See, it is a bicarbonate ion. The bicarbonate compound. The bicarbonate compound with the help of the coenzyme biotin, the enzyme pyruvate carboxylase convert the pyruvate to oxaloacetate. Okay, the pyruvate into oxaloacetate. Then this oxaloacetate is converted into phosphoenol pyruvate. For this conversion also, the magnesium is an important molecule because the enzyme carboxylase, that means phosphoenol pyruvate carboxylase enzyme, requires the magnesium. At the time one Guanosine triphosphate, okay, not a ATP, the GDP, guanosine triphosphate, the another energy-rich compound is phosphorylated and it converts into guanosine diphosphate, GDP. Then oxaloacetate is converted into phosphoenol pyruvate. It is a direct process. But at the time of conversion, it needs here two different ways. If pyruvate is obtained directly from the lactate, it converted into a phosphoenol pyruvate within the mitochondria. But the pyruvate directly involved in the conversion of the glucose, or the pyruvate directly involved in the gluconeogenesis process, that time the pyruvate not directly converted into phosphoenol pyruvate. That means the pyruvate converted into oxaloacetate. This oxaloacetate not converted directly to phosphoenol pyruvate, because this oxaloacetate again converted into malate, because it needs a reducing enzyme NADH. The NADH is necessitated because the NADH is normally present in the cytosol. That NADH regulates the glycolysis process. If the NADH are not utilized again in cytosol, the glycolysis process is automatically stopped, so that the oxaloacetate is converted into malate. Then the way it is converted into malate means the oxaloacetate cannot directly transfer to the cytosol, because the oxaloacetate transportions are not present in the mitochondria. So it converted into malate, then malate transferred to the cytosol. In cytosol, the malate again converted into oxaloacetate, then oxaloacetate converted into phosphoenol pyruvate through PEP carboxylase. These are the steps. This step is clearly explained how this pyruvate molecule converted into phosphoenol pyruvate. Here you see the pyruvate. If the pyruvate directly involved in the gluconeogenesis, the pyruvate which synthesized in the cytosol is directly transferred to the mitochondria. In mitochondria, the first enzyme, the pyruvate carboxylase. Again, I tell you, the pyruvate carboxylase is a mitochondrial enzyme. It only present in the mitochondria. So during the gluconeogenesis, the mitochondria and also cytosol, both cell ion is needed. Okay, so the pyruvate carboxylase convert the pyruvate into oxaloacetate. Here the carboxylation. When carbon dioxide is added here. Why the carbon dioxide is added? The added carbon dioxide helps the activation of the enzyme, the pyruvate carboxylase. It is the activating enzyme. Okay, so the pyruvate converted into oxaloacetate. Then the oxaloacetate, with the help of mitochondrial malate dehydrogenase, it converts the NADH into NAD plus. It converts oxaloacetate into malate. Then the malate transported out of mitochondria to the cytosol. In cytosol, the malate converted into oxaloacetate with the help of the enzyme cytosolic malate dehydrogenase. Here the NADH is again utilized, because during the glycolysis, the pyruvate, the formation of pyruvate, one molecules of NADH is utilized. That utilized the NADH is now reformed through the gluconeogenesis process. Because during this process, if it is not regenerated, the energy synthesized process, the glycolysis is not occurred. Because why means the NADH is an regulating compound, regulating redox couple, which present in the cytosol. So the malate is converted into oxaloacetate with the help of cytosolic malate dehydrogenase, and the oxaloacetic acid with the help of carboxylic phosphoenol pyruvate, cytosolic phosphoenol pyruvate carboxy kinase enzyme. Now the decarboxylation reaction is occurred. When carbon dioxide molecule is removed from oxaloacetic acid, this decarboxylation helps to formation of phosphoenol pyruvate from oxaloacetate. If pyruvate is not a precursor for phosphoenol pyruvate synthesis, or pyruvate is not a precursor for gluconeogenesis. At the time, the lactate is synthesized in human body through anaerobic fermentation or during the rigorous activities or during the exercise. Our muscle cells convert glycogen into lactate. Then lactate converted into pyruvate. The pyruvate conversion, the lactate dehydrogenase enzyme is needed. In lactate dehydrogenase enzyme itself forms a NADH. Here, the NADH is produced in these steps alone. The lactate to pyruvate, the formation of lactate to pyruvate alone, the NADH is formed. So, the NADH is the amount of NADH in the cytosol is maintained here. So, need not to convert the pyruvate into malate here because the phosphoenol pyruvate may directly synthesize in mitochondria itself. After the synthesis, the phosphoenol pyruvate is transported to the cytosol. Okay, the pyruvate, the first enzyme pyruvate carboxylase, will convert pyruvate into oxaloacetate. Then the oxaloacetate to phosphoenol pyruvate with the help of mitochondrial phosphoenol pyruvate carboxylase. Here, one thing you see, the mitochondrial phosphoenol pyruvate and also cytosolic phosphoenol pyruvate carboxykinase. Both the enzymes do the same function, but different location. Both enzymes do the same function, but different location, different cellular places. These both enzymes are controlled by different genes which present in the nucleus. It is controlled by nuclear genome. At the time of which molecule is present in that cell, for that these enzymes, these genes are activated and produced, whether the phosphoenol pyruvates are synthesized in the mitochondria itself or it's synthesized in cytosol, depending on the precursor, depending on which type of precursor is involved. Okay, this carboxylation and the decarboxylation process, the carboxylation and the decarboxylation both are helps to formation of phosphoenol pyruvate. The carboxylation helps to activation of carboxykinase enzyme. The decarboxylation process helps to formation of phosphoenol pyruvate from oxaloacetate. Now we see the second bypass reaction of glycolysis process, that is the conversion of fructose 1,6-bisphosphate to fructose 6-phosphate. Normally in glycolysis for the fructose 6-phosphate to fructose 1,6-bisphosphate conversion, the enzyme phosphofructokinase is involved. This phosphofructokinase is irreversible enzyme. That enzymes are not converted in reversal manner. So the new enzyme is needed. That enzyme is called fructose 1,6-bisphosphatase. This fructose 1,6-bisphosphatase helps to transfer of one phosphoryl group from fructose 1,6-bisphosphate, especially from the first carbon atom. Phosphate group from the first carbon atom is removed. Here, not the formation of ATP. This process is called catalysis. Mere transfer of inorganic phosphate. One inorganic phosphate alone is transferred from the first carbon atom of fructose 1,6-bisphosphate. Then it converts fructose 1,6-bisphosphate into fructose 6-phosphate with the help of enzyme fructose 1,6-bisphosphatase. Yeah, it is this enzyme to the catalysis process. It is a catalysis. Okay, not ATP formed here. It is a second bypass step. In third step, conversion of glucose 6-phosphate into glucose. Normally in glycolysis, from glucose to glucose 6-phosphate, the enzyme hexokinase is involved. This enzyme hexokinase is not function in reversal manner. So it is a third bypass reaction of gluconeogenesis. For the conversion, the glucose 6-phosphatase, this enzyme is needed. The glucose 6-phosphatase enzyme is not present in the blood, in the brain cells. This enzyme needed, it is one of the magnesium activated enzyme. So these enzymes are only present in the liver and also hepatic cells. So that the gluconeogenesis process occurred only in the liver or renal cortex of human, not in all cells. Our muscles and the brain cells Even in our blood, J2 does not contain these enzymes, so that the glucose 6 phosphatase only activated or only present in the our liver cell. So if the lactate or pyruvate which needs or which undergo the precursor for this gluconeogenesis, that are directly transferred to the liver. In that liver, it converted into glucose through these steps. Then it transported through the bloodstream to the needed cells. Okay. In this process also, the glucose 6-phosphate to glucose, this process also the magnesium dependent process. Here, one catalysis process is needed. Here also not ATP is formed. The catalysis, only the inorganic phosphate alone is transferred from the glucose 6-phosphate to glucose. Yeah, it is a sequential reaction of gluconeogenesis. What are the sequential reaction? First, pyruvate with the help of the bicarbonate ion and one ATP molecule, it forms a oxaloacetate. Here the two molecules of pyruvate is involved, so that here I mentioned the two molecules. Then the oxaloacetic acid to phosphoenol pyruvate. And the third step, this red color is mentioned. It's a irreversible process which occurred only in glycolysis. The blue color reactions are only takes the reactions which I mentioned in the blue color that takes place in both glycolysis and also gluconeogenesis. The phosphoenol pyruvate is converted into 2-phosphoglycerate, 2-phosphoglycerate converted into 3-phosphoglycerate, and 3-phosphoglycerate converted into 1,3-bisphosphoglycerate, and 1,3-bisphosphoglycerate is converted into glyceraldehyde 3-phosphate. These steps are reversal of glycolysis in the same manner with the help of the same enzyme. The glyceraldehyde 3-phosphate to diethylacetone phosphate, then the both one molecule of glyceraldehyde 3-phosphate and one molecule of diethylacetone phosphate is converted into fructose 1,6-bisphosphate. Now, again one is reversible process. The fructose 1,6-bisphosphate is converted to fructose 6-phosphate and it leaves one inorganic phosphate. And the fructose 6-phosphate is directly transferred to the glucose 6-phosphate with the isomerase enzyme, and glucose 6-phosphate is converted into glucose. It is one of the irreversible processes. It occurs only in the glycolysis. If we see the overall summary, the two molecules of pyruvate is converted into one molecule of glucose. It needed 4 molecules of ATP, 2 molecules of GDP, 2 molecules of NADH, and 2 molecules of hydrogen ion, and also 4 molecules of water. But it produced 1 molecule of glucose, 4 molecules of ADP, and 2 molecules of GDP, and 6 inorganic phosphate, and also 2 NAD. The deoxidized compound. Generally, this gluconeogenesis is an energetically expensive process, but it is a very essential process. If gluconeogenesis is not occurred, the cells could not obtain the energy during the fasting and also the non-energy process. For example, in between the meals and some of the times. The glycolysis and gluconeogenesis are regulated reciprocally. Yeah, these both are regulated by so many enzymes. For example, the enzyme fructose 1,6-bisphosphatase and the fructose phosphatase, fructose phosphate kinase, and also fructose 1,6-bisphosphatase. These enzymes are regulated whether we undergo the glycolysis or gluconeogenesis depending on the molecules which present in the cell. Let me see the summary of the gluconeogenesis. The overall summary. In glycolysis, the glycolysis is a multi-process step which convert glucose from the 3-carbon compound, example alanine or lactate. The 7 steps are same. In glycolysis and also gluconeogenesis, because these 7 steps are reversible reaction. The 3 irreversible steps also present in gluconeogenesis. Okay, that we see in the pyruvate to phosphoenol pyruvate and the glucose 6-phosphate to glucose 1,6-bisphosphate to glucose 6-phosphate and the fructose glucose 6-phosphate to glucose. These are irreversible process. Then formation of 1 molecules of glucose from pyruvate to 1 molecules of glucose occurs 4 molecules of ATP, 2 molecules of GDP, and 2 molecules of 2-NADH. It is more expensive process, but it is essential one. In mammals, especially the gluconeogenesis mainly occurred in the liver and the kidney, and also brain. Liver and kidney, it converted glucose. That glucose is used by brain, muscles, and the erythrocytes. Okay, so I hope you are well understood about this gluconeogenesis. Okay, animals cannot convert the acetyl-CoA derived from fatty acid into glucose because the plants and the microorganisms can. Animals cannot convert the acetyl-CoA from the derived fatty acid. The gluconeogenesis and glycolysis are reciprocally regulated to prevent wasteful operation. Okay? Because whenever the glycolysis needed, it undergo the glycolysis. Whenever the cells cannot directly obtain the glucose molecule from our meal, that time our cell undergoes a gluconeogenesis that are regulated by the enzymes and also a gene. I hope you are well known about the gluconeogenesis. Do you have any doubt? You feel free comment in the chat box. Okay, welcome.